Hello everyone and welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to study about our next analytical technique, that is ion exchange chromatography. In the previous lecture we have seen various type of chromatography technique, that is thin paper chromatography, HPLC, gas chromatography, column chromatography. So in this particular lecture, we will going to study about what is ion exchange chromatography. Since the name suggests ions, so in this type of chromatography, we will going to talk about ions, that is anions and cations. So ion exchange chromatography or ion chromatography, it is a process that allows the separation of ions and polar molecule based on their affinity to ion exchange. So in this type of chromatography, it is a process that allows the separation. Here also separation is taking place because chromatography, the basic idea behind the chromatography, or you can say principle behind chromatography, is separation. So in this also separation is taking place of ions and polar molecules. So separation of ions and molecules that are polar in nature, they are separated on which basis? On the basis of their affinity towards the ion exchanger. Okay, all these terms we will going to study one by one. The principle of separation is thus by reversible exchange of ions between the target ions present in the sample solution to the ion present on ion exchanger. So this is the definition. If you can see, ion exchanger chromatography, it is a process. Which type of a process? That allows the separation of ions as well as the polar molecule. On which basis? On the basis of their affinity to the ion exchanger. Okay, so let me elaborate this ion exchange chromatography. So there are differences that are found in charges, charge density, or you can say the distribution of charges on the surface of different molecule. Every molecule have different types of charges. Charges, not charges, it's charge. Okay, so these differences in charge properties of different molecules, they are taken into account, and further what they do, they separate the other identical molecule. So in this ion exchange chromatography, it is a powerful technique that is used to separate two proteins which are very similar to each other but different from each other in having one charged amino acid. So you can note it down somewhere. So this type of chromatography, in this separation of, where you want to do a separation of amino acid, suppression of amino acid, I'm writing it in short, AA, amino acid, or not amino acid, you can say separation of protein, separation of proteins which are very similar, in nature. Means they are similar to each other, means here two proteins are taking place, so both the two proteins are similar to each other. But what is the difference? But the different from each other in having one charged amino acid. So what is the difference? So difference in difference from each other in having one charged amino acid. Okay, so this is clear. So this ion exchange, again I am repeating, it is a very powerful technique used for separation of two proteins. Those two proteins are very much similar to each other but different to each other in having one charged amino acid. Okay, so this is again a definition. So in ion exchange chromatography, solutes are retained on an ion exchanger due to their reversible interaction with oppositively charged groups on ion exchanger. Okay, now let us understand this with the help of the diagram. So this is a whole layout of ion exchanger chromatography. So ion exchanger chromatography, they have a chromatography column. In this, sample mixture is placed. Then they have a elution buffer in the charged column, as you can see this place. Okay, so after and low salt elution buffer, so this side is a low salt elution buffer, and whereas this side they have high salt elution buffer. So if you can see this chromatography, they have a sample mixture. Okay, so as the ion exchange chromatography take place, so as you can see in the second diagram, the separation is taking place. Firstly, they act as a, in this I'm just marking here. So if you can see this place, it is a type of a mixture, sample mixture. Due to this ion exchange chromatography, separation is taking place. As you can see, green color is separated. Then again further ion exchange chromatography, again the purple one is separated, and again the last one is the orange one is separated. So as you can refer this definition here it is written now that separation of ions or polar molecule based on their affinity to ion exchanger. So if you can relate this definition based on their affinity to ion exchanger, so you can relate to this. Based on the affinity, they are separated. Means first green one is separated, then purple, and then orange. Okay. Now if you see that here we have used two terms. Okay. First is ion exchanger and second is cation exchanger. So ion exchanger is what? Ion means positive, cation means negative. So negatively charged, as you can see, sorry, I have just told you reverse, that ions is a negatively charged, and whereas cation is are the positively charged. So if you can see this diagram, so this diagram is negatively charged analyte, that is anions, and since they are negative in nature, if you can see this negative sign, so they attract the positive one, so attracted to the positive surfaces, so they attract the positive surfaces, whereas this side, this is a positively charged analyte, that is positive, and they attract the negative. Means we all know that positive attract negative. Okay, negative attract positive. So in this way the attraction and separation take place. Now let's move further to exchange to understand what is the detail of ion exchanger and what is the detail of cation exchanger. So in this process, two types of exchanger, that is cationic and anionic exchanger can be used. If you see the first one, that is the cation, so cation possess negatively charged group and these will attract positive cations. So cations, they are positively negatively charged group, and what they do, if you can see this, they are positively, okay, again I think I've just a little bit confusion is there, so, because of the negative and positive. So cations are the negatively charged and anion is positively. I am just writing it here. Okay, so there must be no confusion. So in the blank, cation and anion. Okay, so these two are the types of ion exchanger. First is cation and second is anion. So if I say cations are negatively charged, whereas ions are positively negative positive. Okay, so they attract which type of uh, ions? So they attract the positive ones and they attract the negative surface. Okay, so cations are negative in nature, whereas anions are positive in nature, and since they are negative in nature, so they attract the positive one, and since they are positive in nature, so they attract the negative ions. Okay, now clear? Now moving further, the cations, they possess negatively charged group and then they will attract the positively charged cations. So what they are attracting, the, as I have written there, they attract the positively charged cations. Okay, these exchangers are also called as acidic ion exchangers. So which type of exchanger they are known as? Acidic type of exchanger. Okay, material, because their negative charge result from ionization of acidic group. So this is again very important. Cation exchangers, they are negatively charged. That is the reason they are attracting the positive charged cations, whereas these exchangers are known as acidic ion exchangers. So in examinations or in MCQs or in interviews, if they ask you about acidic ion exchanger, so don't get confused. It is nothing but it is a cationic exchanger. So they are, why they are, again this question is there in examination, in interviews also. I'm just marking there only, so you can just take a screenshot of this. Sorry. Okay, so I'm just marking it here only. This question in examination, they generally ask in examination that why cationic exchanger, why cationic exchanger, oh, why these cationic exchanger are known as acidic ion exchanger. So the answer for this is because their negative charge result from ionization of acidic group. Okay, so this is the answer for this question. Now moving towards the ionic exchanger. Ionic exchanger, they are positively charged group that will attract the negative charged ions. They are known as basic ion exchanger. Since cationics are known as the anionic acidic ion exchanger, so they are known as the basic ion exchanger materials. Ion exchange chromatography is the most often performed in the form of column chromatography. So it is very much similar to what we have studied about the column type of chromatography. So it works on that principle, and these ion exchange chromatography is most often performed that however, there are also a thin layer chromatographic material that work basically based on the principle of ion exchanger. So if you have studied my previous slides or lecture based on the chromatographic technique, then you can understand this technique, that is ionic exchanger. So they basically worked just like column chromatography or thin layer chromatography. The basic principle of thin layer chromatography is based on the principle of ion exchange. Okay, now let's understand what is the working principle of ion exchange chromatography. So if you see the working principle, so this form of chromatography relies on the attraction between positively oppositely charged stationary phase known as ion exchanger or analyte. So so this form of chromatography, they lies on the attraction between positive charge, that is negative and positive. and they are known as ion exchanger that we have already studied, and analyte. The ion exchanger basically contain a charged group covalently linked to the surface of an insoluble matrix. So ion exchange basically what they contain the charged group, and these charged group are linked covalently on the surface of the insoluble matrix. So the charged group of the matrix can be positively or negatively. So the charged group of the matrix could be positive also and could be negative also, depending upon the material, or you can say molecule. When suspended in aqueous solution, and further when you suspend the charged group of matrix in an aqueous solution, so they are surrounded by the opposite charge. Okay, if the matrix have positive charged, so they will be surrounded by which charge? The negative, the opposite one. And when the matrix, when you suspend the in a aqueous solution, that this matrix, if it contains a negatively charged, so they will attract the opposite charge, that is a positive charge. So in this ion cloud, ions can be reversely exchanged without exchanging the nature and properties of the matrix. So in this, what they are exchanging, they are reversibly exchanged without changing the nature and properties of the matrix. Okay. Now this is the instrumentation of ion exchanger that we have studied. Now what is the working principle? So this is the instruments, or you can say parts of ion exchange chromatography. The first one is eluent, second is pump, third is injector, then is your column, then suppressor, and then last one is conductivity detector. So these are further you can see instruments or parts, you can say, that are used in ion exchange chromatography. So let us understand what is a pump. So in ion exchange chromatography, pump is considered to the most important component in the system, okay, which has to provide a continuous flow of element through the IC injector, column, or detector. So this is the most important part of, or you can say component of IC, that is ion exchange chromatography. Then comes your injector. So injector, since the name suggests injector, so the sample is introduced in various ways. So the simplest method is to use a injection valve. Okay, so you take an injection and you put your liquid sample, and you can with the help of this injection you can directly inject the liquid sample, or you can say sometimes solid sample needs to be dissolved in an appropriate solvent, make it a liquid, and you just directly inject it. Okay, so injector should be provide a possibility of injecting the liquid sample within a range of 0.1 to 100 ml of volume. So if you see injector, so injector is basically used to inject the sample should be in a liquid form. If you are using a solid, then you use a solvent and you dilute it, make a liquid, and then you inject it. What is the volume? So ranging from 0.1 to 100 ml volume can be used in the injector. Then comes your columns. So depending upon its ultimate use and area of application, the column material may be stainless steel, titanium, glass, or inert plastic such as PEEK. So the column can vary in diameter ranging from 2 mm to 5 cm in length. And what is the basic used? So basic used is for the normal analytical purposes or high speed analysis, or you can say sometimes preparative work. So the guard column is placed anterior to the separating column. So this serves as a protective factor that prolongs the life and usefulness of the separation column. So here the separation generally take place. And column could be of stainless steel, column could be of plastic such as PEEK, column could be of titanium glass. Okay, now comes the detector. So electrically conductivity detector is commonly used. And what is the data system? So basically data system is needed, and in this the information is displayed in the form of in the screen that is connected to the computers. Now comes the application of ion exchange chromatography. Now if you look at the applications of ion exchange chromatography, so the first application of ion exchange chromatography is an important use of ion exchange chromatography is routine analysis of amino acid mixture. So as we have studied about the definition also, they are very much important in the separation of two protein molecules which have different amino acids. So they are basically used in the routine analysis of amino acid mixture. The 20 principal amino acid that from the blood serum or from the hydrolysis of proteins, they are separated and they have a clinical diagnosis in use in clinical diagnosis. This is the most effective method for water purification. So in water purification also ion exchange chromatography has a huge application, or this is one of the most effective method. Complete deionization of water or a non-electrolyte solution is performed by ion exchanging solute cations or hydrogen ions and solute anions for hydroxyl ions. This is usually achieved by method used for softening of drinking water. Okay, where you want to make your water purified, or you can simply say if you want for the purification of water, ion exchange chromatography is used. In the analysis of products of hydrolysis of nucleic acid, in this way information is gained about the structure of these molecules and how it relates to their biological function, such as carrier or hereditary information. So in this also ion exchange chromatography is used. So where it is used? In the hydrolysis of nucleic acid. Gelating resins are used to collect traces metal from the seawater. So all these have the applications of ion exchange, but the most important is amino acid mixture, analysis of amino acid mixture. Then it has a clinical diagnosis also. Used in clinical diagnosis, they are very effective and one of the most used method for water purification. So it has a various range of applications in different field. Then comes the advantages of ion exchange chromatography. So if you look at the advantages, so it is one of the most efficient method for separation of charged particle. So whenever you want to separate a charged particle, this technique or chromatography is one of the most efficient method. It can be used for almost any kind of charged molecules, including large protein, smaller nucleotides and amino acids. So it is very much all used for any kind of charged molecule separation, like large molecule or small nucleotide and amino acid. Ion exchange is used for both analytical and preparative purposes. So it is not only limited to analytical technique or processes in the laboratory. No, ion exchange can be used for both analytical and preparative purposes. The analytical uses being the more common, but if you compare where they have a more advantages, so they so in analytical use they are more common. Then inorganic ions can also be separated by ion exchange chromatography. So if you want to separate inorganic ions, so this type of chromatography can be used. So these are certain advantages of ion exchange chromatography. So if you have advantages of any technique, so there are certain limitation that is there. So if you see the limitation of ion exchange chromatography, so only a charged molecule can be separated by this technique. So if you want to separate a charged molecule, then this technique is very much important or very much useful. But if you want to separate any you can say uncharged molecules, they cannot be separated by using this technique. Buffer requirement is always there. Okay, so they required buffer, so because without buffer they cannot separate. So these are certain limitation that is there with the ion exchange chromatography. So that's bring to the end of the lecture. Let us quickly understand what are the types of question can be asked in the interviews. So in interviews or while giving any examination, this question is very much important. What do you understand by ion exchange chromatography? Or they can ask you on which principle ion exchange chromatography works. So you must be knowing the principle as well as the definition. Then they can ask you to differentiate between ion exchanger or cation exchanger. So you must be aware of a cationic exchanger and anionic exchanger. Then with working principle of ion exchange chromatography, this is the must question. Every time in examination you get this question: what is the working principle of ion exchange chromatography? And we have studied different types of chromatography. Ion exchange is one of the important ion type of a chromatography that is there in your questions paper, in your interviews. Apart from ion exchange, HPLC and thin layer chromatography, these three have a huge demand in your examinations. So you must be aware of the working principle of ion exchange chromatography. Now instrumentation, of course, for your understanding, you must be aware of the type of instruments or components that are there in ion exchange. They can ask you up to which volume you can use in the injector. So ranging from 0.1 to 100 ml volume you can use. Okay, and which type of column can be made from which type of material? So stainless steel, glass, and sometimes plastic, that is PEER. And so these instruments or notation can be asked in MCQs or in interviews. And yes, of course, the applications are always there. So suggest any two applications where ion exchange chromatography is used, or they can ask you the advantages. And whenever there is an advantages, of course limitation are always there. So these are certain questions that can be asked in the interviews. I hope you have understood the lecture. If you have any doubt, any query related to the lecture, you can leave your queries in the comment section below. In the next lecture I'll be answering the queries. You can follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn. And yes, subscribe ITLS Academy and visit our website, that is www.itlsacademy.com, for further information. And this is our helpline number, that is 7080833450. If you have any doubt, any query related to any course, so you can simply call on this number and get updated. So again, thank you students for watching. Don't forget to like, share and subscribe ITLS Academy. You can get ITLS Academy app from Google Play Store. So you can go there and upload this app of ITLS Academy and get a latest update related to the lectures. Thank you.